that I'm going to give you is a sort of a general view on the second brain of the human body, taking into account that what we have in the bowel is really a second brain generated by the complexity of connection of a number of neurons comparable to those we have in the spinal cord. So it's a very remarkable system. And this system, as pretty much like the central nervous system, can get affected by several knocks or injuries or damages, whatever, and can generate changes which can be neuroplastic in a way, and the possibility that these neuroplastic changes may save the function or somehow preserve some reflex ability. And in this context, I will give you my interpretation of this topic. Now, let me start with the first molecule. For me, number one, most important of everything else, which is this poor patient, as you can see from the bowel, slightly distended, very thin, very skinny. You can see here the prominence of the bone and the pelvis and the chest here. And this was a 29-year-old female girl coming to our attention because she had the nausea, vomiting. She could not eat anything basically, and because of this, she had first diagnosis of IBD, and of course there was no evidence of IBD at all. Secondly, she was crazy. She was so crazy to have 2 psychiatric admission in a psychiatric hospital in northern Italy. We are not talking about the southern component of our country, we are talking about northern Italy, and this is really, I can tell you, surprising for me and for you, and I'm glad that you laughed, because this was exactly my first reaction when they said the story of this girl. So nothing like that. This girl had a severe disorders, functional disorders of the gastrointestinal tract. So the sad point is starting my presentation on neuroplasticity is exactly this one. Nobody had thought about the possibility that there is something wrong in the innervation or the control system in the bowel of this girl. So what is wrong in this system was wrong, of course, the motor component, the absorption, the secretion, and of course there are several other components that we are now acknowledging as integrative of these basic physiological patterns, such as the immune response, the changes of microbiota, blood flow, and also very recently the increase in intestinal permeability, which would allow translocation of bacteria in other systems and other organs, therefore generating infections, sometimes fatal for these poor patients. And the very important component which is underlying all this is the digestive nervous system, which is very important, controlling all this function, motility, absorption, secretion, and the novel aspects of the gastrointestinal tract. So what I'm talking about the gut innervation with its intrinsic and extrinsic component. I will come to a moment to the intrinsic component. The extrinsic component is represented by the vagus nerves, and you can see here different colors representing the afferent component. You can see here starting from the esophagus or the stomach, particularly the upper part of the stomach, the fundus, and going through the parasympathetic component of the vagus up to the nodus ganglion, and the nodus ganglion to the wall of floor, if you wish, of the fourth ventricle, where the nucleus tractus solitarius is, and relaying with some neurons, and from these neurons to the dorsal motor vagus, nucleus of the vagus, and where neurons are giving off a long neurites innervating the upper gastrointestinal tract, but also some other parts of the digestive system. So this is pretty much a very short summary of the parasympathetic component represented by the vagus. Then there is also a sacral component, more importantly innervating the lower part of the gastrointestinal tract, and there is in the middle the sympathetic component giving its origin by the neurons located in the lateral column from T1 to T8, and from these neurons there is a first synapses on the prevertebral ganglia. You can see here the celiac ganglion, the superior mesenteric ganglion, the inferior, and also the pelvic ganglion. So all these are relay for the first neurons located in the spinal cord. And from this, of course, another neuron is innervating the digestive system, providing the sympathetic component. But the recent view of the physiology of the digestive innervation has been centered on the innervation supplying the gastrointestinal tract in its intrinsic component, so the enteric nervous system. Why I'm saying that? Because the enteric nervous system is a very remarkable collection of neurons characterized by plexuses located between the two important components of the muscle, the longitudinal layer and the circular layer. And you can see here the several projection coming out from the myenteric ganglia, also referred to as Auerbach's plexus, and also the submucosal ganglion, or referred to as Meissner plexus, according to the name of the researcher who first discovered the presence of these neuronal aggregates. So this is a long knowledge of the enteric nervous system. And very importantly, data indicated that the number of neurons is pretty much the same of the spinal cord, as I said before. So 5 to 10 to the 8 or 1 to 10 to the 8, depending on humans or experimental animals, or in general in mammals, which is not so far away from the largest collection of neurons we have in the central nervous system. So this is a very important component controlling the vast majority of the gut function, those that I just presented you in the first slide, and those that are wrong in the patient that I just showed you at the beginning of my presentation. And very importantly is this system is also able to interact with the parasympathetic and sympathetic components. So from that early evidence to the very last understanding on the enteric nervous system, which is now referred to as brain in the gut, a real little brain, or brain, if you want, present in the wall of the gastrointestinal tract, we know now that there are as many transmitters as those contained in the central nervous system. Probably more, according to my good friend Marcello Costa. This cocktail of messengers contained, as you can see in this very nice representative example by Mario Colucci in my laboratory, showing this complexity, pretty much something like 20 functionally distinct classes of enteric neurons. So we talk about, for example, intrinsic primary afferent neurons, which are those neurons capable of detecting the information generated by the distension, for example, or chemical coming to the lumen of the gastrointestinal tract. But also the brain in the brain, as my good friend Marcello Tonini used to say, referring to the interneurons, but also the effector neurons, so the motor neurons, largely investigated by most of us dealing with the enteric nervous system. There is here my good friend Marcel Jimenez. He just generated very important evidence on inhibitory junction potential, which are exactly an expression of motor neurons, which go through an inhibition of the function generated by that specific neurons on the muscle, for example. These neurons are connectedly integrated in various reflexes. We all know peristalsis, but also even more complicated reflex system, or if you want, a nerve pathway, such as those controlling migrating motor complex, the secretomotor reflexes, and also, of course, many more vasomotor reflexes, for example, and so on. This system is pretty much, as I said, integrated with the parasympathetic and the sympathetic component, but remember that it is able to work independently from any other supply of the central nervous system or peripheral nervous system, whatever you want. So it is generated, is completely individualized in such a way that it is able to work autonomously. There are also some other important players. Of course, it's not only neurons, such as enteroglial cells or glial cells. Very importantly now, there some important work that has been generated by Michel Néon-List and other groups in the world in this respect. And also, of course, the interstitial cells of Cajal, which represent a very important component in, let's say, transducing the effect of neurons. But what is really remarkable in my understanding as a clinician with an interest on the internal nervous system and in general the digestive innervation is these neuroplastic properties. What does it mean, neuroplastic properties? It means that there is a neuroplasticity going on exactly as we know in the central nervous system, also in the anterior nervous system. We can define this accordingly to morphological or functional changes, adaptive changes, adaptive mechanisms which are going on in the anterior nervous system again, or self-maintenance or repair program which are activated by noxer or injuries or any stimulus which is able to activate this neuronal component which are programmed to repair or self-maintenance. And of course there is a large interplay of environmental factor, but also genetic factor, which may be responsible for these changes. So we were taught when we studied at the university that the nervous system, any kind of nervous system, is composed of cells devoid of a replicative potential. They last for the entire life, and this is something that has been defined by such outstanding investigators such as Giulio Bizzozzero, but also Santiago Ramón y Cajal used to say that. So not, we are not talking about Roberto de Giorgio, which is the last investigator in the world, but we are talking about very important historical people. Bad thought. This is wrong, as the vast majority of the important acquisition given by science are going to be regenerated and refreshed by new understanding, and this is the new understanding. So these many different multicolor neurons present in the central nervous system or even in the anterior nervous system named or defined as stem cells. These are, of course, already committed towards the neuronal component, but these are pretty much stem cells, which may be activated by any time, and they can replace, for example, dying neurons or the degenerated neurons, which is extremely surprising. So there is nothing everlasting. There is something that can be replaced and in appropriate situation. So how can occur that this cell can be activated and the stem cells can regenerate or take the place of something that are altered? And what are the causes altering the morphofunctional integrity of this system? Well, there are several factors which have been demonstrated to have a role in that. For example, genetic factors, many genetic factors. I would spend the entire afternoon talking about this area. I wouldn't do that, of course. Degenerative disorders, particularly Parkinson's disease and Alzheimer's disease. Unfortunately, we don't know exactly what is really degenerative in the enteric nervous system or even in the peripheral nervous system, as pretty much like what we know in the central nervous system, which are much better defined patterns identifying degenerative neurons. And also the loss of enteric neurons, because when we count neurons, we know that, for example, in congenital or acquired neuropathies, there is a reduced number of neurons. For example, diabetes is just one of these, but I could list many other conditions in addition to that. And the role of infections or intraluminal noxae, for example, such as gastroenteritis, but also changes in gut microbiota, but also the very emerging topic of food allergies, which is a very important one now in the field of gastroenterology, and never completely well defined, I would add also. In my modest experience, there is nothing that we can do to define exactly what is a food allergy. But anyway, then of course the inflammatory component, such as the activation of the immune system in the inflammatory bowel diseases, but also in less overt conditions, such as the irritable bowel syndrome, I will show you very few examples, a short example of what I'm talking about, and in inflammatory neuropathies as well. So the large spectrum of sensory motor disorder can be covered by inflammation as a factor contributing to the alteration observed in these conditions. And of course, symptom generation. So let me elect to investigate with you and show you some data on degenerative disorders, particularly pointing of my attention on some recent data that we are contributing also in this recent moment in our laboratory to investigate with Parkinson's disease, according to the recent evidence from, again, Michel Neunlist group, generating very important preliminary results on these, in this respect, in this condition. So there is a large array of conditions such as Alzheimer and also Parkinson's disease, but many more conditions affecting both the central nervous system and the enteric nervous system. In this condition, the degeneration is much more acquired than what we know when we look at the patient without any Alzheimer or Parkinson, but there is only something that is going bad in his or her bowel. So this is a paradigm also to investigate neurodegeneration. As I said, Parkinson is one of these. What is this? Is a very important condition. I don't want to spend much time on that because I'm sure that you are familiar with the Parkinson's disease, which is a motor disorder, but characterized also by an important autonomic dysfunction. And just to give you an example, there are important cardiovascular abnormalities in these patients, urinary and sexual abnormalities, dysfunction of the thermoregulatory system and skin changes, of course, associated with that, also pupillary abnormalities, and likewise sleep and respiratory disorders, which are, of course, very tremendously impacting the quality of life of these patients. And last but not least, of course, the gastrointestinal disorders. I wouldn't take all this long way to get to the point. The gastrointestinal disorders are certainly number one in this, in generating these abnormalities and impacting on quality of life. The biomarker of disease, that's why it is very important, is represented by these Levy bodies. We know that when there are Levy bodies, we are talking about a Parkinsonian component characterized by aggregates of phosphorylated alpha-synuclein inclusion in the central nervous system and as well as also in the peripheral nervous system and enteric nervous system. In patients with Parkinson's disease. So these are the major GI dysfunction characterized by frequent and debilitating symptoms. They can affect the whole gastrointestinal tract. The main complaints are represented by hypersalivation, up to 78% of patients, according to this review by Pfeffer in Lancet Neurology. But I would like to focus your attention on dysphagia, which is a high dysphagia characterized also not only by the involvement of the smooth muscle component of the esophagus, but also the striated component of the esophagus, up to 97% of patients, and constipation, which can occur in as many as 80% of patients, and usually is a severe form of constipation, not so easily responding to classic laxative. And gastroparesis can also occur, which is, of course, generated sometimes by the use of dopaminergic treatments. As you all know, dopamine is a potent inhibitor of gastrointestinal function. Any function of the gastrointestinal tract is inhibited by the dopaminergic system. So this could worsen even symptoms. Of course, in this condition, the dopaminergic supply represents a treatment, and it is not only per se a component of worsening or symptoms by itself, but it can contribute pharmacologically speaking. And the pathophysiology is largely unknown. There are several possibilities to investigate Parkinson's disease. One of these is, of course, the human tissue procurement, which is not so easy, of course, and the experimental models. But most of these experimental models in our experience and in our hands do not really generate the real Parkinson's disease that you can investigate, of course, having the possibility to use human tissues. So this is very important to me to stress the concept that human tissues are very important for unraveling the alteration, the abnormalities underlying Parkinson's disease. This is a very old experience by Qualman and Wakabayashi, studies during the 90s, 80s, showing the Lewy bodies and Lewy neurites, as I said, the hallmark of the Parkinson's disease, detected in the enteric plexus of patients with the Parkinson's disease and involvement of the gastrointestinal tract. In this case is the esophagus, but also the colon, as you can see here, particularly in this study. These are very old study. That's why the picture are quite terrible, I would say. But just to give you the idea that the concept of a Parkinson's disease, involvement of the gastrointestinal tract in this condition was not unknown by most of investigators. Unfortunately, we cannot procure full thickness biopsy. Not only that, but also the recent evidence generated particularly by this investigator, Braak, pointed out on the possibility that it is very important to investigate the gastrointestinal tract for a very peculiar reason. The first thing, the most important one is that the ENS lesions identified at an early stage of the disease can be so early to precede the abnormalities or the neuronal loss observed in the substantia nigra, which characterize the pathology of Parkinson's disease. Of course, I'm talking about the central nervous system. So this is very important to stress the concept that the gut could be a sort of a quoted window to investigate Parkinson's disease and, of course, the patient affected by this severe condition. Also could represent not only a way to investigate the pathophysiology, for example, of constipation, but by itself predict the future or the evolution or the outcome of this condition. So, practically speaking, what Braak thinks about this condition is that a wide array of injuries, for example, factors like toxins or pathogens or even changes of gut microbiota may activate the molecular machinery generating the abnormalities of synuclein, which is the base for the formation of Lewy bodies in the enteric nervous system, in the submucosal and menteric plexus. This would be a signal for the diffusion of the condition of the disease up to the central nervous system. And there is recent evidence that the synuclein can even be secreted by the synaptic side and therefore could be transmitted up to the central nervous system. We don't know exactly this clearly, this aspect, but definitely they could contribute to the pathophysiology of the transmission of the disease from the periphery to the central nervous system. And therefore, there is a vast area of investigation in this respect, not only from the gut side, but also from the synaptic component of the Parkinson's disease. Basically speaking, what the researchers think now is that Parkinson's disease is something starting from the gut as a synaptic disorders and diffusing up all the way up to the central nervous system. The interest generated by this Braak's hypothesis led this group of people, particularly Pascal Derkinder and Michel Neulist, to envisage the possibility to use common, simply applied mucosal biopsies to investigate the submucosal plexus and therefore to investigate Parkinson's disease, which is extremely excellent way how to procure tissue for analysis. And this is what I'm talking about, very simple. That's what we do also in our laboratory now. We just pick the submucosa. You see here, this is a classic, the classic device how to obtain mucosal biopsy. This is the little blood spot, and this is the forceps to obtain the tissue. And you can see where does it go farther down when we picked up the tissue at this level, the submucosal plexus. So this is the area, basically. And this is the biopsy obtained. You can see here the mucosa here, and the forceps separating the mucosa from the submucosa. What they obtained in patients with Parkinson's disease is the evidence of the presence of alpha-synuclein. So they obtained the presence of levoneurites in 72% of the patients, so 21 out of 29 Parkinson's disease patients with levoneurites in the submucosa, in the submucosal plexus. And by the way, this is NF220, which is a marker of the enteric nervous system, a general marker of the enteric nervous system. And this is very interesting because you can see here in this part of the slides, which it is not the length of the disease or the duration of the disease, if you wish, but the intensity of the pathology, which correlates with the reduced number of neurofilament reduction, which is basically this marker here in the enteric nervous system. So it means that the degenerative disorders is associated with the strongest Lewy pathology. So they come up with the possibility that the levoneurites in the submucosal plexus might be a new biomarker for Parkinson's disease, just looking at the gut. We generated also an interesting review article published a few years ago, where we defined this, in Parkinson's disease, the brain spill its gut, because we wanted to bring up this issue, as I said previously, that the enteric nervous system could be a nice way how to investigate, in general, neurodegenerative disorders such as Alzheimer's, and not only Parkinson. We are also generating similar results. This is our equipment. This is a microscope. This is the biopsy. This is what we obtain in terms of, the submucosal biopsy. So basically reproducing the technique, and this is our evidence of PGP 9.5, which is a general marker of the enteric nervous system, and VIP immunoreactivity. So this is pretty clear that the feasibility of the technique is there, and we can use this to investigate conveniently the enteric nervous system. Of course, we don't know how much this is comparable to the enteric plexus, but definitely could be one way how to investigate the alteration occurring in any degenerative disorders, including, of course, Parkinson's disease. Now, end of the story for degeneration. Let's move on to inflammation, particularly in these three settings. As I said before, particularly I would like to concentrate on IBS and inflammatory neuropathies. And before getting that, I would like to just refresh you what we know about the neuroimmune interaction in the gastrointestinal tract, and why is that? Just because the gastrointestinal tract is the largest human lymphoid organ. There are as many lymphocytes as those contained, for example, in the spleen, of course, in the length of the gastrointestinal tract. It contains 80% of immunoglobulin produced by lymphocytes, producing lymphocytes, and is in a constant state of physiological quoted inflammation. It's constantly activated by the number of factors coming from the lumen. And of course, this is the reason why there is a lot of attention now on intestinal permeability. I'm sure that you are also investigating this in functional bowel disorders, as we are doing now. Not only that, but also there is a bidirectional interplay between neurons and the immune system. The neuropeptide receptor, for example, are expressed on immune cells, and vice versa. The immune cells release a variety of mediators in response to neural stimuli, but also the immune mediator receptors are expressed on enteric neurons, and enteric and sensory nerves respond to immune stimuli. So there is a wide array of potential represented by representing the neuroimmune interaction. So this component fits very well into the spectrum of gut sensory motor disorders and inflammatory changes characterized by very commonly identified disorder, labeled probably in an improper way as a functional GI diseases, which means nothing in my knowledge, but definitely that's what has been applied, and it is still currently used, which in this perspective, in the inflammatory perspective, means a minimal inflammatory component. As you can see here in this represented example, these red spots are mast cells infiltrating the mucosa, and therefore these are mainly represented at the mucosal level, which are thought to evoke sensory motor changes. This is the base of the iceberg, and at the other part of the spectrum, at the opposite of the spectrum, there are these severe dysmotility disorders characterized by marked inflammatory infiltrate in the anterior nervous system. As you can see here, an example of this, what we call now enteric ganglionitis. So it's a dense packed infiltrate within the enteric plexuses. Typically, the muscle are in this context are not involved by the inflammatory component. So it's very restricted to the nerves. That's what we call, as I said, inflammatory plexitis or inflammatory ganglionitis or enteric ganglionitis. So let's take this first in this presentation. We are here. I'm here presenting the data from my good friend Giovanni Barbara. You can see that Professor Barbara demonstrated the presence of a dense infiltrate of mast cells. Again, this is an example of what I'm talking about. The red spot are the tryptase positive mast cells. Tryptase is one of the general markers for mast cells, such as CD117 or c-KIT, pretty much the same, or kinase. We can use also kinase. And you can see here the dense arborization of nerves in the mucosa. And the vicinity of the mast cells, again here in the overlay, in red, and to the nerves supplying the mucosa. So this is a quite close contact between the mucosa, the mucosal nerves and the mast cells. And you can see also the electron microscopic data showing that these mast cells are degranulating and therefore releasing their component, their granules close to the nerves. And also this is another example talking about or showing again this close contact and the release of the mast cells granules or intracellular bodies basically close to nerves. And these morphological aspects that I just showed you correlate with abdominal pain severity score and with a frequency score. So both abdominal pain severity and frequency are correlated with the vicinity of mast cells to nerves. And particularly, if we would like to just give you a more detailed information, is less than 5 micrometer of vicinity in terms of clinical impact of pain, of abdominal pain. So these are very relevant data. Giovanni went on to demonstrate that these mast cells release a vast cocktail of substances such as tryptase, proteases, histamine, serotonin, which can contribute to activate nerves and, of course, generate possibly dysfunction in this respect. My part was to investigate the very important component represented by these patients, which are not commonly observed in our clinical practice, but definitely they are severely affected by severe dysmotility, and particularly looking at the changes occurring in the neuromuscular layer. In the inflammatory perspective, again, this is the evidence of an enteric ganglionitis. You can see here the dense packed infiltration of inflammatory cells limited to the enteric plexus. These are distributed pretty much to CD8, CD4, T helper, and T suppressor. The long way to over time of this condition may lead to severe dysfunction characterized by marked neurodegenerative changes. So the inflammatory insult is such to evoke neurodegenerative changes. And as I said, this is not very common, but when we see a picture like that, it is possible that an immunosuppressive treatment may be indicated for these patients. We know that there are evidence clearly from a clinical side and also from what is published by the literature that is not only effective this, as a strategy, as a therapeutic strategy, but this because probably we don't know exactly at the time when we treat these patients if the neurons are completely gone. there is evidence that when the inflammatory component is completely gone, there is also very few neurons left in the myriad plexus. And in this regard, it's important to show that previous researchers, previous investigation have clearly demonstrated the presence of an aganglionic component in these cases with a long-standing story of inflammation in the myriad plexus. In addition to the inflammatory component represented by lymphocytes, we have an immune activation characterized by neuronal antibodies in patients with severe dysfunction, particularly pseudostructure. Usually the paraneoplastic component is more than the idiopathic form, but there are some patients with no evidence of paraneoplastic syndrome. And the presence of these autoimmune component is represented by immunoglobulin G to U proteins. Therefore, that's the label of anti-U or ANA1, which means nuclear neuronal antibodies type 1, according to the definition given by Van der Lennon and this group at Mayo Clinic. So this is what we obtained from taking blood of one of these cases, one of these patients with the lymphocytic ganglionitis and the presence of autoimmunity with autoantibodies. These antibodies label very nicely the neurons in the central nervous system or in the anterior nervous system, both in the submucosal plexus or like here, or in the myriad plexus, in the jejunum and colon of course, an experimental animal. We cannot use the human tissue, we have to use experimental animals. We can also use Western blot to identify the molecular target of these antibodies, and this is the UD component of this protein. So this is pretty much what we can be realistically done with the patient with an with a suspect of an inflammatory neuropathy, or demonstrated by the lymphocytic ganglionitis that I just showed you before. But the question is that is this humoral component able to contribute to neuronal dysfunction, such as the neurons that we just showed you affected by or insulted by the lymphocyte that I just showed you a couple of slides before? The answer is yes. One of these mechanisms is represented by autophagy. We tested this in a neuronal cell line. In patients with pseudostructure, we identified at least that 1/4 of this patient carrying anti-neuronal antibodies of the type that I just showed you before. And when we apply this antibody to the cell line, to the neuronal cell line, these activate the LC3, which is a marker of the autophagy. And this marker of autophagy is associated with a phosphate ligand pathway, which is very nasty for the cell, leading to neurodegeneration. So this is a possibility that a neurodegenerative component may be activated by these anti-neuronal antibodies. But there is not only a component of lymphocytes and immunoglobulins. There are also other cells of the immune system which can contribute to severe dysmotility. One of these is represented by eosinophils. You can see here several eosinophils in the myriad plexus. This is a pediatric case. This is an adult case, and you can see here the expression of IL-5, which is a chemokine with a very important chemoattractive component to eosinophils. The very interesting point here is that the eosinophilic infiltration is not associated with neurodegeneration. Very minimal neurodegenerative component. The highest is the highest expression is a severe dysfunction, as shown from a clinical standpoint. Again, not only lymphocytes or eosinophils, but also mast cells. This is as I showed you before for the IBS, also for the severe dysfunction of the gastrointestinal tract, may be activated by an infiltration of mast cells. And indeed, these are all listed function characterizing the features of the mast cells. They are sentinels of the immune system. They respond to a wide repertoire of stimuli. They release bioactive substances substances I just mentioned you before. The activation is accompanied by neurally mediated secretory and motor reflexes. And also there are distinct subtypes of mast cells. Not all mast cells are similar. There are different types of mast cells according to the area of the gastrointestinal tract and the layer of the gastrointestinal tract that you are dealing with. So this is a study that we presented some years ago with Anna Carino, Professor Malgelada, and Fernando Aspiros. These are the series of patients with severe gastrointestinal disorders and mast cell infiltrate. They are all characterized by mast cell infiltrate. Of course, these data are old. We are now having new data from more component coming from more data from new patients from this group, and at that age they were represented by patients with pseudostructure, one case, and 3 patients with inertia coli, and also 7 patients with severe functional gastrointestinal disorders / enteric dysmotility. So these are basically cases where I see, from the histopathological point of view, a dense infiltrate of mast cells in the muscle, as you can see here and here, or surrounding the myenteric plexus. As you can see here, this is the myenteric plexus with the presence of mast cells surrounding, and also the heavy distribution not only in the muscle but also again within the myenteric plexus. So it's a sort of mastocytes, if you can let me use the term mastocytosis, quoted mastocytosis of the gastrointestinal tract. So very peculiar, very unusual, and leading us to investigate this further. So I'm coming to the last part of my presentation after this long way that I just try to take you around with the several possibility of dysfunction and the neuroplastic potential of the internal nervous system. The future perspective is the possibility to use a very minimal invasive laparoscopic surgery, and with the use of full thickness GI biopsy. As you can see here, this is the armamentarium, and also the approach that the surgeon has with this laparoscopic surgery. The least manipulated is the gastrointestinal tract, and better is the best is in terms of the resultant ischemia, in terms of reduced ischemia, of course, with the laparoscopic surgery, and particularly reduced inflammatory infiltrate activated by manipulation. We have also guidelines now how to use this biopsy, and these are 2 papers derived from a consensus conference with Charlie Knowles and myself leading this group of people, expert in neuromuscular pathology, and also coming up with a London classification, which is basically a way how to correlate the neuromuscular pathology with the clinical phenotype. The endoscopy is also progressing a lot. This is the endocytoscope through which we can get full thickness biopsy. You can see this is the serosa pulling out this component, which is represented by muscle and mucosa. You can see here the mucosa, the muscle, which can be viewed directly with this technique, and also, of course, with the histopathological examination, for example, investigating nitric oxide synthase and many other transmitters important for gastrointestinal regulatory roles that they have. This is a very recent paper coming out from Carla Cirillo and Peter Vandenberg in Jan Tack's group, where they investigated the electrophysiologically the submucosal neuron obtained with this, similarly with this technique, basically, something like that. Or if you want, very similar to what I showed you before for the Parkinson, you can see here the activation, the different type of neurons as identified by UCD, which is a general marker of the enteric neurons, and also by another marker. And you can see here the activation by neuron 1, here represented by the green line, and neuron 2 here represented by the red line. So this is possible to obtain this distinct activation by the MPP, which is a cholinergic agonist, and also by serotonin, for example, by these 2 neurons with eliciting, for example, action potential. So the future will be to investigate further this with minimal invasive approaches. In the way that we are now, which is leading to new potent pharmacological approaches for gut dysmotility and sensory disorders. Of course, we don't need to have such a fancy research going on, but definitely I think that there is a role in the future for stem cells. My personal view, of course, Professor Tapar is a good friend working in London in this field of research, and he is using stem cells committed towards either neuronal or glial component and to replace this in aganglionic gut. He's a pediatrician, so therefore the main phenotypic disease that they're looking at is Hirschsprung disease, which is an aganglionosis of the gastrointestinal tract, as you know. How they do that is a very conservative way. Again, this is a microbiopsy of the fetal gut. You can see that this microbiopsy are already provided with neurons, as indicated by TUJ1, and SMA, which is a muscle, so there is very little muscle here, but you can see here several nerves, and not only that, but also that these are committed towards several lines, as indicated by this marker, SOX10 GFAP+, which is a glial oriented line here, and also neuronal components that are coming up with this SOX10 GFAP minus, which means that these are neurons in the way to be differentiated into mature neurons, basically. Very interestingly, if we engraft these isolated neurons in a aganglionic hindgut, for example, of an experimental model, in this case is sheep hindgut, you can see that they reproduce over time a myenteric plexus. You can see here the circular muscle, the longitudinal muscle, and this is a nice myenteric plexus as identified by a general marker of the enteric nervous system. And you can see here again the myenteric plexus here with a production of nitric oxide synthase or vasoactive intestinal polypeptide, so two transmitters of the gastrointestinal tract. So this is a potential of future application. What does it mean to have a translational research going on in this field? Meantime, we wait for a new prokinetic or a new probiotic or prebiotic or whatever you want. So I come to the conclusion of my presentation. I hope that not give you too many info as I usually do, but this is my part of my enthusiasm in this field of research. A wide array of factors or conditions play a pivotal role in enteric neuroplastic changes, particularly those underlying various GI diseases. Of course, a general sentence, but still very important to me in a translational perspective, is that the better knowledge of the mechanism involved in neuroplasticity may enable the development of targeted therapeutic strategies. For example, one of these could be the use of neurostem cells, but there is also now recent evidence indicating that some classic serotonergic receptor may have a very important role in neuroprotection, for example, in eliciting neuroprotection. This is, for example, the recent Michael Gerson research area where 5-HT4, the same receptor targeted by prucalopride, can be a neuroprotective pathway for neurons, at least in experimental animals. We are investigating that in humans. So an acknowledgement to my colleagues. You can see here Giovanni Barbara for the study on IBS, my teacher, Professor Corinaldesi, the second teacher, but similar teacher, of course. I don't want to say number one, number two. They are both number one for me. Vincenzo Stangellini, the group of research here, many numerous, many young people and many more coming up with difficulties in generating money for them. The important collaboration with Anna, Carolina, and Fernando and Professor Malgelada here, but also in many other respects with other groups for the other ongoing research. And a thing to my good friend and mentor, Professor Marcello Tonini, who was an outstanding researcher and an outstanding pharmacologist, and he passed away a couple of years ago. And unfortunately, I'm still missing him and his mentorship in this field of neurogastroenterology. The data, most of the data that I just showed you, coming from our laboratory, and many more that I wanted to have to bother you to show you, came from discussion and intellectual interaction with this man. We hope that you will come to Bologna. Not only Fernando, he must come, and Dan, of course, but many of you. We are organizing this meeting, the joint international meeting of neurogastroenterology and motility, organized by Vincenzo, Giovanni, and myself. And of course, the three societies: American Motility Society, European Society, and Functional Brain Gut Axis group for the three societies leader in neurogastroenterology in now. And thank you very much for your attention.